Hello and welcome to this video in the series on integrated disease management in cereals. In this video we're going to look at the question of exactly which parts of the crop canopy do we need to protect and when. So it might seem odd to start a video series about integrated disease management by thinking about the crop and its physiology. But a key point of IPM is that we're balancing between the productivity of the crop and the impact on the environment. So it's important that we know exactly what it is we're trying to achieve. So we're going to look closely at the physiology of wheat and barley and which developmental phases are sensitive to the effect of diseases and which need protecting. So let's get started. This is video number 1 in the series on integrated pest management, science and practice, focusing on disease control in cereals. And we're going to focus today on a question about which parts of the crop canopy need protecting and when, because a key part of IPM is that we only apply control measures when they're required. And we're going to use the example of wheat and barley as two contrasting systems. So the first step is to think about two questions. What determines grain yield, and how and when does foliar disease affect yield? So yield is formed by photosynthesis. So on the left-hand side, this is a cross-section through a cereal leaf. Light energy from the sun is arriving on the left, it's penetrating into the leaf. It's intercepted by chlorophyll, which sits within chloroplasts, which are the little green dots within the leaf. And the photosynthesis drives the absorption of carbon dioxide, which is fixed in carbohydrate, and water through transpiration is lost in the process. If we then look on the right hand side what this does, if we imagine we went to a wheat crop and every week we cut down the wheat crop and took away everything that was above ground, and we put it in a huge oven and we dried it right down and drove off all the water, what we'd be left with is the amount of dry matter, in this case measured in tons per hectare. And we do that through time. What we can see first of all is during February and March there's not much sun and there's not much crop canopy to intercept it, so growth is very slow. But from early April onwards the sun angle improves, day lengths get longer, there's more crop canopy to absorb the solar radiation, and crop growth goes into this period of very rapid expansion. Initially that's growth of leaves and stem, and also as the stem enlarges, the stem starts to be filled with what we're calling here stem reserves. So these are short chain carbohydrates, usually fructans, that are quite readily moved about in the plant, and those get deposited in the stem as it expands. Then we reach a point when leaf and stem growth is complete, the ear emerges, and the ear starts to grow. And then after flowering, that ear is able to accept carbohydrate and start to fill the grains. And we can see from flowering onwards, the growth of the crop is growth of the ear, which is predominantly the grains, and that's supported in two ways. First of all, by real-time photosynthesis during grain filling, so carbohydrates going from the leaves to the grain, but also by a reallocation of the stem reserves into the grain until those stem reserves are exhausted. And there can typically be between 2 and 4 tons of those stem reserves available, so it's a pretty significant amount of dry matter. So that brings us then to the question of what it is that's limiting yield. And crudely, there are 2 possibilities. If we look on the left, we've got the amount of source of dry matter that we have available. So that's determined by the amount of solar energy that the crop receives and the proportion of that solar energy that's intercepted by green tissue and converted into dry matter. So that's our dry matter source. On the right hand side, what we've got is our dry matter sink. So how much capacity have we got to store the carbohydrate and to some extent the protein that we're producing? And that's determined by the number of ears per metre squared, the number of grains per ear, and the potential size of those grains. So if we imagine a crop in which we had far more source available to produce carbohydrate than we had storage sink capacity to fill, then our yield would be limited by sink, and we need to focus very strongly on how we can protect and improve our sink capacity. If, on the other hand, we had a crop which had loads of sink capacity but less source capacity, so we had more ability to store than we have to produce, then we need to focus very carefully on how we can protect and improve our source of carbohydrate. And this really is going to guide how and when we decide to protect the crop from disease. So let's look at those 2 components in a bit more detail. So source capacity is the amount of photosynthetically active radiation intercepted by green canopy, and we measure that in megajoules of photosynthetically active radiation. And that's multiplied by the radiation use efficiency, which is the number of grams of dry matter we can produce per megajoule. So the radiation use efficiency is predominantly determined through plant breeding and through crop nutrition. Foliar diseases can have some effect on radiation use efficiency, but their predominant effect is through the amount of light that can be intercepted by green canopy. So now let's look at that component in a bit more detail. If we look at the crop on the picture on the left, if we imagine we went into that crop, we marked out a metre square of the crop, we went in with scissors and we snipped off every bit of leaf area that we've got, and we flattened it out, and we measured the amount of green leaf and stem that we've got. In the picture in the left, what we would find is we've got twice as much leaf tissue as we have ground area, so 2 metres squared of leaf and stem per metre square of ground. We know that because we've got physiologists who are daft enough to do that. In the middle picture, if we did the same exercise, we'd find that we've got 4 metres squared of green tissue per metre square of ground, and the crop on the right, we'd find we have 6. So let's just imagine for a moment that you're a ray of sunshine, which I know you are, and you're shining down onto this crop pretty much as we see it here. In the crop on the left, there's quite a good probability that you would hit the ground rather than green leaf tissue and therefore be wasted. In the crop in the middle, even though we've got 4 units of green area, there's still some probability that you would hit the ground instead of green tissue, And the reason for that, of course, is that quite a lot of those leaves are overlapping each other rather than sitting sort of neatly side by side. But by the time we get to a GAI of 6, then it would be pretty unlikely that a photon of solar energy would strike the ground. It's highly likely instead that it would hit green tissue and therefore be usefully turned into dry matter. And we can quantify the relationship between the amount of green area we've got and the amount of solar radiation we intercept through a relationship called Beer's law, which is unfortunately nothing to do with beer, but was originally defined to describe how light is absorbed in a liquid. And it turns out that we can think of the crop canopy as a kind of thick green soup that absorbs light. So we can see if we have a green area index of 2, bottom left, and we follow the dotted line up, we're intercepting just over half of the available light. At a green area of 4, we're intercepting about 80% of the light. And by the time we get up to a green area of 6, we're up to about 90% plus of the light being intercepted. So this is a pretty good measure of our source capacity, how much carbohydrate we can produce. And we'll come back to disease and the effect of that a bit later on. But first we need to think about the sink capacity. So sink capacity determined by the number of ears per metre squared, the number of fertile grains per ear, and the potential grain weight. And this is where we come across the first big difference between wheat and barley. So if we look closely at the side of a wheat ear, what we see is that each spikelet, and we're showing one spikelet in the image on the left, can have up to 9 fertile florets in which grains can form. So these are fertile flowers. And typically there'll be between 4 and 6 fertile grains per spikelet in wheat. If we compare that with barley on the right, barley only has 1 or 3 spikelets which are forming fertile florets, depending on whether it's a 2-row barley or a 6-row barley. So there are 2 important consequences from that. The first is that barley inherently has less potential to create fertile grains. And the second, which is equally as important, is that wheat has great flexibility to adjust its sink capacity according to growing conditions, whereas barley is far more limited in its ability to adjust its sink capacity. If we then look at the effect of those processes on the balance between source and sink, and here we're just thinking about the healthy crop, we haven't really introduced disease yet, and this is data from the wheat and barley growth guides, we can see that the amount of source between wheat and barley in a typical crop is not that much different, but perhaps a bit smaller for barley, although the effect of a difference between 5, 6 or 7 GII units on interception is not that great. But there is quite a big difference in the sink capacity as measured by the number of grains per metre squared. So what we've got in a healthy crop in wheat is that typically the source and the sink are in close balance. We've usually got enough sink capacity to store whatever amount of carbohydrate we can produce. In barley, that is often not the case, that we have more source capacity than we have sink capacity to store it. So that has big implications for disease management, which we'll come to now. So let's introduce disease into this system. This set of photographs here happens to be for Septoria tritici over in the west of England in 2022, where we flew a drone over a set of wheat plots pretty much every 2 weeks from the peak of the canopy size through to senescence. And if we just follow down the plots outlined in green, we can see at the top around the 2nd of June the canopy was healthy. On the 17th of June, being well protected in this case with fungicide program, the canopy was still predominantly green and still green right through to the 23rd of June. By the 1st of July, it's starting to lose green area substantially. By the 8th of July, it's senescing. If we now follow down the series of plots which were untreated, outlined in yellow, we can see at the top the canopy is green, but by 23rd of June it's lost a substantial amount of its green area and is already starting to senesce. So if we now relate this to what that does to light intercepting capacity, this is a fairly typical curve of green area index up the vertical axis, plotted against time and growth stage along the bottom. So we can see as the upper canopy is expanding that the disease has rather little effect on canopy size, mainly because the canopy is kind of outgrowing the epidemic and producing new leaves before the disease can destroy them. But then at a certain point those leaves stop being produced and the epidemic catches up and starts to reduce the green area index of the canopy. So we can see the difference between the healthy and the diseased. And if we then relate that back to Beer's law, we can see what effect that's having on our source capacity through light interception. So let's imagine, looking along the bottom, that we grow a really massive thick canopy that has 9 units of green leaf area, and we lose 1 unit of that green leaf area to disease. We follow those dotted lines up, we follow them across to the left. We see that because we were kind of on the plateau of Beer's law, we're losing rather little light intercepting capacity. If on the other hand we've got a canopy of 5 units of GAI, then losing 1 of those units of green area is starting to have quite a significant impact. We're losing about 10% of our intercepting capacity. And by the time we get down to a canopy of 2, if we lose 1 GAI unit of that, then we're having a really quite big effect on fractional interception of light. So we can think about this in 2 ways. First of all, we can think about this as a kind of time series. We start with a canopy, we lose more and more of it to disease, we lose more and more of it to natural senescence. The impact of a unit disease gets bigger and bigger as time goes on. And we can also think about it as how big a canopy we grow in the first place. So you could say, oh well, so what we should do is we should grow a really big canopy and then we can afford to lose a bit more of it and it won't really have much of an impact on yield. But to do that, we'd have 2 effects. First of all, we'd be spending a lot more on nitrogen than we need to spend from an economic point of view. And secondly, excess nitrogen encourages certain biotrophic pathogens such as the rusts and powdery mildew, making it more likely that we'd lose more of the canopy. So we have to bear in mind that the disease severity that we see as a percentage is a percentage of a variable amount of canopy and therefore doesn't relate directly to the impact on yield. It's the remaining green area that relates to yield. So now we've understood source and sink, we need to think about when those two things are being determined and therefore when they need protecting from the effect of foliar disease. So along here we have a timeline of crop development and growth stages, and we can see there's a period of canopy expansion from emergence of the crop, which ends when the ear is fully emerged at growth stage 59, and we then enter a period of canopy duration from full canopy expansion through to senescence. And those two green boxes, as we've seen, are determining our source capacity. Then in orange, we've got the components of our sink capacity. So production of tillers and tiller survival is occurring almost through emergence through to just after foliar emergence, with some differences in these time periods between wheat and barley, but broadly similar. We have a period of spikelet and floret initiation and then survival that determines the number of grain sites per ear. And we have a period of ear growth. So this is the structure of the ear growing, which determines the number of cells there are in each grain, which is related to the potential size of that grain, how big it could get if there was adequate source capacity. And that happens sort of in a few weeks either side of ear emergence. And then after flowering, we move into a period of grain filling. So we can see that these different components are occurring at different times, but the critical question now is, which of those processes are sensitive to the effects of disease? And that turns out to be really different between wheat and barley. So first of all, if we think about wheat, and we're thinking about the effects of foliar diseases, so it's rare for foliar disease in wheat to have much of an effect on canopy expansion. It's rare for foliar disease to have much of an effect on ears per metre squared. We quite commonly see a small effect in wheat on grains per ear, and we see a big effect on canopy duration, particularly if the epidemic is severe. So if you recall, we start out in wheat with source and sink being in close balance, but here the effect of disease is to reduce the source far more than it reduces the sink, and therefore a disease crop is source limited, and we need to do everything we can to protect that source. On barley, the situation is almost the reverse. So we do see small effects on canopy expansion, but we typically see quite big effects of foliar disease on ears per metre squared and grains per ear. And even quite small amounts of disease can cause quite big effects on those sink components. And sometimes we even see an effect of fungicide increasing those sink components even when there's no visible disease in the crop, although that effect is quite hard to predict. And we also see an effect of foliar disease on canopy duration. So the effect on sink is bigger than the effect on source, and we already had a sink that was tending to be smaller than the source even in a healthy barley crop, and this has exacerbated that. So we now have, typically in a diseased barley crop, something that's quite severely sink limited, and what we really need to do is to protect the formation of sink during the period when tillering, tiller survival and floret survival are being determined. We can now think a bit more about which parts of the crop canopy need protecting as well as when. So in the case of wheat, we can work out the amount of light intercepted by different plant parts during the period when those mobile stem reserves are being accumulated and during grain filling when the source capacity is being determined. And we can see predominantly it's the upper leaves of the canopy and the ear which are contributing most to yield and therefore need most protection. We did consider whether we could produce a similar diagram for barley, but decided in fact it would be misleading because it really isn't the source capacity during those later stages of the crop that are predominantly detecting or determining the effect of disease on yield. So to conclude, which parts of the crop canopy need protecting and when? So in wheat, disease management is about protecting the dry matter source. So we need to protect the upper leaves from when the flag leaf is fully emerged at growth stage 39 through to flowering to protect the accumulation of carbohydrate stem reserves, and we need to protect those upper leaves from flowering through to natural senescence to protect dry matter accumulation during grain filling. Now, we're going to need to plan that protection and apply protection by whatever means right through from the previous autumn when we decide on the variety and sowing date. But the ultimate aim is protection of the crop canopy later in the season. In barley, the situation is fundamentally different. We're trying to protect dry matter sink formation by protecting tiller formation and survival to maintain ears per metre squared. And we're trying to protect ear growth, particularly during booting, in order to maintain fertile florets and potential grain weight. And surprisingly, 2- and 6-row barley behave rather similarly because although 6-row barley has more grains, they're typically smaller and the crop typically still remains sink limited. And also winter and spring barley behave rather similarly, but the period of sink formation is very compressed and short in spring crops and is particularly sensitive to early disease. So I hope this has defined what we're aiming to achieve. How we achieve these aims is dealt with in subsequent videos in this series. And for further reading, please take a look at these papers.